So before I start, I'll just kind of give you a high-level kind of overview of how I think about the stuff that I was doing and working on. I would say I'm a neuroscientist, and I think a lot of people interested in the brain kind of come in to neuroscience really interested in the mind. So how people think about things and solve problems, you perceive and act in the world, and also when stuff goes wrong, when you have neurological diseases and stuff. And then a very common kind of methodology is to record the brain while you're doing all kinds of stuff, right? So some stuff we saw yesterday, like during working memory, and also during diseases, and basically try to find these neural correlates of cognition or disease. But I really kind of also wanted to approach this by thinking about what are the components in the brain that gives rise to these signals, like really just treating the signals that record from the brain as like physical measurements from a physical system, and then see what you can kind of derive or infer about the brain or the system that it was recorded from, right? So today I'll mostly be talking about sort of the link between these two things. So the first half, or not even the first half, but the first conceptual half will be sort of the analysis methods that we're working on how to kind of more think about these brain signals more mindfully and decomposing them into stuff that makes sense just by looking at the data. And then also the complementary direction, which is how do you synthesize or model brain signals from mechanistic variables that or that experimentalists have found with like real animals, right? And by this, I really just mean like models and simulations. And by analysis, I'll get into what is called the inverse problem. So kind of fitting models to data. Okay. Yeah, so I guess Pascal had this like explicit instruction for me to be didactic because there's sort of this wide audience of wide expertise. So I'm going to keep it like really simple at the high level. And there's really like 3 takeaways that you should kind of remember. And there's sort of like 3 slides that I'll point you to, this being the first one, which is, the first one is this ideology, and I'm kind of like giving my propaganda of what I really believe people should be doing more, which is what I already described to you, treating brain signals as just measurements from physical systems. I mean, we can think about them like using dynamical systems framework or, as computations through dynamics or whatever, but first and foremost, I think they're just like physical signals from the brain, which is also a physical and biological system. Okay. And second thing, I'll talk about a little bit about the parameterization of power spectra, neural power spectra. Some of you probably know FOOOF. I think Peter, yesterday you were looking for me. That's me. Yeah. Or it's not me, it's Tom, but I'll talk a little bit about it. And the last thing, I'll tell you a bit more about some of the new stuff that I've been doing, tubing in, kind of combining machine learning for neuroscience applications. Okay. And feel free to ask questions anywhere during the talk. Yeah, so the first thing, the ideology, so I'm gonna have you this picture here, but I'll kind of draw a metaphor to something else, which is this picture. Does anyone know what this is? Yes. So this is it's a absorbance or transmittance spectrum, specifically an IR spectrum or infrared spectroscopy. So plotted on the x-axis is like frequency or wave number, and on the y-axis is transmittance. So it's this thing that you do in organic chemistry. You shine some light at a thing, and then you measure what wavelengths of lights are transmitted or absorbed. So you can do all sorts of crazy stuff with this because each of these peaks corresponds to some particular chemical bond or organic chemical bond in this thing, which is just like a long hydrocarbon chain. Here's another example of bromomethane. So it's like this thing, bromine attached to a methane group. And the energy of the vibration in this thing gives rise to these peaks in the spectra, right? And I think this is very cool because you can just look at the spectra and people have figured out, and you now learn this in sort of undergrad organic chemistry, like what each of these peaks in the frequency spectrum corresponds to, and you can attach them to physical things, right? And so what I'm thinking about is how do we do this for brain signals and the stuff that we can derive out of like the frequency spectrum of the brain signals that we measure. Right? And this is not a new idea, it's not my idea. There was this like really big review paper, for example, where they proposed to integrate all these biophysical findings from experimental results where like different ion channels, for example, have different delayed constants, which gives rise to all these different frequencies. And there's like, you know, thousands of these such mechanisms. And I just want to do this all on the computer, right? Like I did my PhD in San Diego, so go to the beach and work on your computer. You can't do that in a lab. So that was what I wanted to do, just to be a computational kind of person. Which is also not a new idea. And a lot of stuff that I'll be talking about kind of are inspired by all these people, if you know them, just computer modelers, try to integrate experimental findings into simulations. So that's the ideology, that's the first important part. And then now I'll talk about how do you maybe measure the spectra of neural signals a little bit more mindfully. Okay, and I didn't pick the frequency thing by accident, right? So I'll be talking about Fourier analysis, and I think most of us here are probably familiar with Fourier analysis, which essentially you have this time series, you have, you know, a basis function of sinusoids, and you can take the dot product for between the signal and all of these sinusoids, and you can get a spectrum like this. And this is a real neural power spectrum. Okay, and you can see like there are different frequencies, and then there's a big peak in the middle already at 10 hertz ish in the yellow color thing, and that's what you call like maybe alpha oscillation. You can plot this like log instead. That's what I like to do. And, you know, again, the peak is very prominent. You can see this, but you can also see that there's other very characteristic features in the data here. So for example, this 1-ref decay, which basically exists for all kinds of neural power spectra. It doesn't matter if you're measuring from inside the brain, outside the brain. Another feature that we were looking at is sort of this bend that transitions from lower frequencies to higher frequencies. And the idea here is basically the frequency spectrum of a brain recording is not all oscillations either. It's got this sort of background stuff. And so today, yeah, this first part I'll talk about basically this background aperiodic spectra, and specifically, so this is sort of like the only math thing I'll show today. Decomposing this thing into these two components. L is the line, which is the red dashed line, which has this like knee and 1-ref component, and then G is just a bunch of different Gaussians centered at different points. That's the peaks that represent oscillations. Most importantly, this is really this package called FOOOF. The people that know it, this is sort of the heart and soul of it is Tom, who was another PhD student with Brad. I wrote maybe like 20 lines of code for this, basically specifically like this one K and F thing, which I'll show you later. But this is work that we did with Brad in San Diego. And sort of advertisement is if you're also looking at neural data or any time series in general, if you're interested, just FOOF it. It's like 4 lines of code. If you have a power spectrum and some frequency, well, you get out of like np.fft.fft frequency power spectra, throw it in there as 4 lines of code, and you get a thing like this. Okay. When you do this, you'll see actually that like there's not so many oscillations in the brain. Typically, there's just 1 or 2 in a given location, and the rest of the stuff can be more parsimoniously explained by this background. Do you have a question?
Yeah, just to understand your FOOF, the black is the real data in this example that you showed?
Yes.
And what is your output?
The output is the red line, which is kind of faint, but it's also invisible here. The red line you can't see is behind it. That's the fit. That's the model fit. And then the dashed blue line is the model fit other than the oscillation peaks. Yeah. So that's the output that you get. I won't talk about the oscillations. You should look up Tom's papers. He has a lot of stuff basically looking at how you could conflate oscillations with this 1/f thing. But today I'll talk about mostly the 1/f and the time constant. So in here, this 1/f is this this chi thing, which is exponent that changes basically the slope of this line. And so to come back to the framework I was talking about before, what can you do with this, right? So once you measure this, what are you going to do with this? So we had this idea. Let's start from like a biophysical-ish model, which is like a super simple model. You forget about all the other mechanisms. You just have a population of excitatory and inhibitory cells, and then they just fire in this sort of asynchronous regime, which really just means random. So you draw Poisson spikes from there. But if you do that and you simulate the field potential or the synaptic currents that they would give off of, you can simulate what the synaptic current looks like, and sort of by first approximation what the recorded LFP or ECOG or whatever looks like. So that's the forward model. Yeah. And then because you have this knob that you can tune between, that's the sort of weighting between the excitatory and inhibitory firing rate or strength, you can adjust them. And when you do that, you'll see that the relative ratio between E and I gives rise to this change in this 1/f thing, okay? And so what you can then do is to go backwards and infer, well, if I have a change in this 1/f, and does it correlate with, yeah, and obviously it does, because that's why I'm showing this. But, and this is like a super simple model, but the real question is, when you apply this to real data, does it give you stuff that also makes sense? Okay. So this first paper in 2017, that's kind of what started me on this whole journey. We showed that basically maybe it does. And then there are a bunch of follow-up papers with better models. So some papers have used spiky neural networks models with, thalamic inputs, and also, yeah, just adding more and more realistic components into the model itself, and shown that basically the gist of it still holds. Then we can do that. You can use this to interpret the signal that you measure from the real brain in all sorts of different ways when they're correlated with. So for example, this interpretation that, balance is changed in autism, for example, or in different diseases like Parkinson, schizophrenia, and, just basically any manipulation you can do to a person or their brain, you can measure and interpret under this light. But the takeaway is that this is now a sort of non-invasive biomarker that you can use for, interpreting physiologically what happens to someone when their brain signal changes, right? Either between days or between different states or between tasks when they're doing task A versus task B. Okay. You can also do this without sort of this forward model. So here, now instead of this 1/f exponent thing, I'll look at this K term, which is basically this characteristic frequency, like when the bend happens, it has this plateau from flat to 1/f. And this corresponds to, so this is like a pseudo-Lorentzian function, and this corresponds to the time constant, the delay time constant of the autocorrelation function in the time series. So if you just take the time series, you compute the ACF, and you measure like the characteristic decay time scale, that corresponds to this frequency thing, right? So we can kind of convert between frequency and time domain interchangeably. But when you do that, you again can measure what this time scale is in different brain regions, for example. So this is an example spectrum from human ECOG recordings, and you see that different areas of the brain that's colored by the different colors have different time scales. So we took this big data set of ECOG recordings from MNI, I think same stuff that you use, Peter, and you can compute a whole brain gradient of timescales. And you see that in this picture, there's some like macroscopic structure in how the timescale changes across the brain, right? And with this, you can tap into a different strand of literature. So this is all in human data, and people have done this in monkeys with like single unit recordings or single neuron recordings that have shown that in monkeys and in rodents as well, that basically as you go from the sensory areas of your brain, so like visual, auditory, sensory, where the input first come in, to the frontal, temporal, parietal areas, the timescales get longer. And the idea is that, these basic primary sensory areas have to react very quickly to sensory stimulus in the environment, whereas the frontal, more cognitive areas have to integrate stuff over a longer time scale. And that's why the activity itself has a longer time scale. So yeah, so basically borrowing from this idea, we can integrate the human data with a bunch of other kinds of data that we had, or the community had. For example, structural data from MRI. You can measure the amount of myelination. The detail is not super important. Basically, the structural measure of hierarchy, cortical hierarchy. So the idea that I was just talking to you about, you can measure this in a different way using just MRI. And if you do that and you compare the two, these large-scale cortical gradients, and you see that they correspond in the way that you expect, right? So what I was saying before, to the far right is the sensory areas, and you have lower time scales, and then as you move towards more these association higher order areas, you get increased time scale. So it kind of checks out. You can also look at, so the Allen Brain Institute has this gene expression database where from brain donors, they were able to measure gene expression of like thousands of genes, all kinds of stuff like synaptic receptors, ion channels, etc. And you can look at the gradient of those proteins across the whole brain, and you see that also these two things correspond. So this marker, this dynamic marker from ECOG corresponds to these structural and sort of mechanistic things that we want in the real brain. And this is sort of the one slide of behavior I'll show, which is the timescale also changes dynamically. So if you put this person in a task where they're expected to remember something in their working memory for some time later, you would expect basically between when they're trying to remember and when they're not trying to remember, their timescale, if the brain is actually supporting this longer timescale integration, during the remembering period, the timescales will get longer. And that's basically what you see in the data as well. So this is the y-axis, the ratio between just the delay non-task period to the remembering period. And the ratio for all of these different areas is above 1. So during task, the time constant is increased in all of these different areas. And what's interesting is that the amount that this increase is for an individual subject corresponds or is correlated with what their overall task performance is. I'm not going to make any causal statements here, but that's just a nice correlation that we found. And there's a bunch of other things in this paper from a couple of years ago where you saw that as people got older, their timescales across the whole brain also decreased, which is consistent with another strand of literature that was showing basically aging-related effects. Okay. Cool. So that's all the stuff you can do. But kind of coming back to this idea of the whole brain gradients is we know a lot of these things, like your brain is not composed of independent systems that are able to independently modulate themselves, right? All things are related to each other. So the gene expression, the expression of all these different genes come together to, for example, give rise to this other gradient in macroscopic organization of what the hierarchy of the brain looks like. And specifically what that means is, for example, these are just two genes. So GRIN2B is an MDA receptor, one of the subunits of an MDA receptor, if that makes sense to you. It's an excitatory receptor gene. GABA is an inhibitory receptor gene. And you see that, y-axis is timescale. So you see that again, timescale correlates with these expression of these different synaptic gene things, right? But it also correlates with a bunch of other genes. So these other 4 genes are, ion channels and whatnot. And more kind of, I guess, concerning is that these different synaptic receptors have different compositions. So a single synaptic receptor could have like 4 of these different subunits, and these different subunits are encoded by different genes. That's the biology of it. So an MDA on top, for example, has 6 of these subunits, and depending on where you are in the brain, the composition of these 4 different subunits or the identity of these 4 subunits are different. Why am I telling this is that it's very complicated you would want a simple story of like, for example, oh, NMDA and a certain brain measure is just correlated. But the messy truth of the matter is that there's a lot of different things in NMDA, and they all correlate with the brain measure or the dynamic measure differently, right? That goes the same with the GABAergic synapses, as well as these, sodium, potassium channels, which are like super important things for brain function. Just for finding an action potential, you need a sodium, potassium channel, right? And different cell types of inventory cell types and whatever, any gene you can think of, it probably correlates, but in sort of unexpected directions. So this is the phenomena of everything everywhere all at once, which is great for biology because that's how your brain works, but sucks for a modeler because then you have to take account of all these things in your model and kind of have to decide, well, I don't really care about this, and sort of justify why, right? So in the last 15, 20 minutes or so, I'm going to tell you about the current work I'm doing in my postdoc. Yeah.
Sorry, I'm going to ask a stupid question. Could you go back to previously to the thoughts you had?
Yeah.
So, right. So the Y scale is the
time scale.
Yeah, it's time. And what's the X scale?
X scale is the amount of expression. So each dot here is one of the locations on the brain. So there's 180 locations or parcels in this thing. You can measure the gene expression in every one of these.
And so how
much is expressed?
Okay, each part. Okay.
And the RNA that you measure is sort of a marker for how much of the protein that's actually there, but not exactly.
Okay. Yeah. Gotcha. Thanks.
Any other questions?
Combine this with any kind of pharmacological intervention, because then you could, instead of having everything everywhere all at once, you could kind of target a specific receptor type.
I haven't personally. I didn't do any of these experiments. These are just data from the internet, basically. But people have, in subsequent papers, looked into stuff like this. Yeah. Both for this timescale stuff as well as the EI balance thing that I was telling you. Cool. So, yeah. So now I'm going to move on to basically the same idea, but kind of amp it up a notch with all these different things combined together. And try to use machine learning to solve this problem. So this is the previous picture, right? We have some model, you can simulate for blah, blah, blah. I just told you, now you have, instead of a single mechanism of VI balance, you want to put a bunch of other stuff together, like the expression of different ion channels or the connectivity in the network. And what you want to do then is encode all of these realistic or real world mechanisms into a computer model, right? And then simulate it, you get data that looks kind of like the real data you might record. If you're familiar with these, the red and blue are the raster plots, so spikes from the network. The black line is the black thing underneath is just the total population fire rate. And then the PSD is the power spectrum of that. What you want to do is that you can do this as much as you want. You have control of what you put into the model, how you simulate the model, and what the data looks like. But what you want actually is to take data that you've observed and somehow put it backwards, right? Meaning, how do you infer, given the real data you have, what changed in the biological mechanism? And as a stand-in, what we're going to say is, well, if you can make the real data look like, or rather, if you can make your simulated data look like your real data, then maybe your model looks something like the real brain network that you have, and you can make conclusions based on the parameters in your model. Okay. So how do you actually do this? The problem, the technical problem is going to become apparent once I describe to you what the model looks like. And the model here I'm using, it's a spiking neural network with sort of biologically the point neurons, so it's not real neurons, but they have real differential equations that describe the voltage dynamics of the single neurons. Specifically, they're called adaptive exponential integrate and fire neurons. So it gets an input and then every now and then fires a spike and then resets. And the spike follows this exponential sort of process. It's a great model because it's kind of simple. It's just two ODEs, but it can, depending on different parameters in the equations, it can give you all sorts of different single neuron dynamics that people have observed experimentally. Okay. So that's the pictures there. Top is real, bottom. No, these are all fake actually, but they're model reproductions of what a real neuron could look like. And you can have different regimes of activity, right? Like just regular firing or bursting, and then with different adaptation time constant, et cetera. So these are the single neuron parameters I want to encode in the neurons. And there are like 10 of these. At the network level, you want to be able to vary things like the amount of excitatory cells and inhibitory cells, for example, what is the strength that they're coupled to, and then, potentially, not topology, topography of the networks, like clusters of excitatory cells coupled with different inhibitory cells, et cetera. So there are 8 or so parameters. And these are just things that I've decided, right? Because you can encode these with arbitrarily more parameters, but I've chosen just, a few more. And then there are things like the actual connections between the neurons, so the synaptic strength, like between E to E, E to I cells, what is the actual conductance, right? So altogether, there are 28 parameters, and all the neurons share the same parameters already, so that's a big constraint. In the real brain, each of these neurons have different values for those parameters, or at least different draws from the same distribution. But here, they're all just fixed to the same constant value. Nevertheless, you have 28 parameters in this network, and changing the value of any one of them changes the dynamics of the network that you simulate.
So when you say clusters, you just mean excitatory altogether in one cluster and inhibitory in another, right? Or
no, so in this particular model, the inhibitory neurons are shared, and then there's this big pool of excitatory neurons in which they're like anywhere between 1 to 20 clusters, which have preferential connections within them.
Mm.
So the probability of connecting with your friend in your cluster is higher than to outside of your cluster.
so these networks are generated. You say, okay, I want some clusters, or do you see the clusters after you've generated this?
You just say like, I want, for example, two clusters.
Mm-hmm.
And given that there are 800 neurons, they randomly get distributed into the 2 clusters. And the number of clusters is a free parameter, so you can go from anywhere between 1 to 20.
Okay.
So so the amount of clustering changes, and the strength of the clustering also changes, because you can then tune how much is the amplification of connectivity between, like, with your friends versus not your friends. Okay. So you put this in a computer program called Brian, and you can simulate. It's just a nice framework for simulating these ODEs, and you can basically, these are 28 parameters. That's the prior range, which I draw from. Yeah.
How big are your networks?
2,000. So 2,000 neurons total, and then depending on the percentage of VNI, it gets distributed. So if you pick one random draw of 28 numbers, that instantiates a single network, and it might look like this. Okay. That's what the spikes look like, and this is the actual simulation from this set of 28 numbers. And then you can compute further downstream stuff like PCA, we saw yesterday, power spectrum. These are all things that basically we care about as neuroscientists. You can look at a different draw of the 28 numbers, and you get a different dynamic in the network. You can keep doing this. You get really crazy stuff like transient bursts that look all different. So I don't know really what to call this. I just gave them random names. You can also get like synchronous oscillations and oscillations of different timescales that involve different assemblies, really super long timescale oscillations that lasts for like 10, 20 seconds with these burst intervals of like, 60 seconds or so. And this basically spans the whole range of your parameters. Okay, and I guess the thing to point out is typically what we would do is as modelers to assume a certain value for maybe 25 of these parameters, fix them because you say, well, I don't care about them, or it's too messy to vary them, and then you just kind of study the parameter regime or the phase portrait of 2 or 3 different parameters, right? But here, if you allow all of them to vary, they give you different stuff. It's not super difficult to imagine, but the difficult problem is then I might actually skip this. The difficult problem is how do you go backwards, right? Given a simulation, you can, for example, measure some statistics that you care about, the inter-spike interval between different spikes so the interval between different spikes for a neuron across the population, that's one summary statistic you can measure. And you can say, I want to correlate that with one of the parameters in the network. So you can make the scale apply. Each one of those dots is one of the 1 million simulations, and it looks like this. So there's some structure, right? And there's a general negative correlation. Measure a different thing, the delay between these bursts, and correlate with a different parameter, the percentage of E to E connections. It looks like that. You can measure the 1 over F, it looks like this, so on and so forth. And these are all things we care about, right? So you can correlate arbitrary measures that you want with one of these 28 different parameters, and you can get a big correlation matrix that looks like this, which is all very significant statistically, but quite, I guess, low effect size. So anywhere between negative 0.2 to 0.2. There's some strong ones that is kind of expected, but overall, everything correlates with everything else. And so the problem is when you have real data, what you want to say is, given, for example, the real data, the real power spectrum looks like this, and I, for example, measure the 1 over F exponent, what are all the, can I generate 28 values that correspond to these parameters that will then give me the simulation that I want? Is that clear? Okay, so that's the inverse problem, right? Yes, that's the inverse problem. Given real data, how do you infer the actual values for these different parameters? And there's a lot of technical challenges, which I'll just like super quickly go through. Massive parameter space, 28 is already really big for these kind of, for the modest neuroscientist. And most of these configurations actually don't give you something interesting. It either goes to silent or gives you these like basically maximally firing networks. And the ones that are sort of reasonable in the sense that like it's something that you might observe in the real data, it's got a large range, like the plot I was showing before. If you allow 28 things to change, they can give you all reasonable things, but span the entire range of the parameter space. So how do you find parameters that's consistent with data, right? So typically what you do, especially if you're a master or PhD student, is you have this model and you basically just type different numbers in the computer for 2 weeks, 2 months I did this for a little bit in my PhD and I gave up, but basically you're typing random numbers until the network matches what the data looks like, kind of, and you have to do some squinting. Because the model is super stochastic and nonlinear, right? So, yeah. But you also want more than a single model that fits the data because, again, the models are stochastic, so on different runs of the model will give you a different result. So, and also different model configurations might give you the same looking data depending on what same means for you. Okay, so that's the problem of degeneracy, basically. So what we're gonna do, how we approach this is to solve this with machine learning. And the way you should think about this is, so you have these circuit parameters. I had this 1 million set of simulations, right? So you have this big matrix of 1 million by 28 features. And each one of those 1 million things produces, you pass it through this function, that's the simulator, and it gives you some data, right? And the data is, you call it X, and you can then choose arbitrary readout of the data. You can take the full time series or a single dimension of, or like a single summary statistic of time series, whatever it is, that number is K, you get this matrix of 1 million by K, yeah, Daniel.
We're just making a grid of the parameters.
In the prior, they're just drawn randomly. So define some prior range in the 1D marginals, you just sample like in this cube. So you have this data matrix, and the goal is basically to find the inverse function of F, right? So your simulator, enacts this thing that given theta, which is the parameters, it gives you X. That's the forward. The backwards, you want this thing G, which given X gives you theta. That's what you want. Such that when you have real data, real X, which is green, you put it in G, it'll give you a real set of values. That's the sort of pseudo-mathematical formulation of this problem. So you want G. Again, quickly, there's some existing options for you can do this. You can use this with just like a feed forward deep neural network. You can try to optimize over this directly, optimize over F directly or G directly if you have gradients. You can do this in a probabilistic formulation. There's an approach called ABC. I'm happy to talk about with people afterwards if you're interested. But we, in particular, Pedro Gonçalves and in Jakob's group, we take this approach of using neural density estimators. So neural networks that basically form probabilistic distributions. Yeah, and I think I have some, and so this entire framework is called simulation-based inference. So you're doing inference based on simulations that you've done in the model. So yeah, that's Pedro. That's the paper that he had from 2020. And in this part, we were just using basically this method to kind of inversely solve the simulator. And this thing is called normalizing flow. And yeah, I want to go through this because this is kind of pedagogical. So I might skip through some stuff later on, but I think this is, if you understand this, I kind of did my job today. Basically, the idea is like you have a bunch of pairs of these simulations, right? Theta and X. And you can draw a number from this prior distribution. So here, the simulator is just 1D, or it is one parameter, and it gives you one dimensional data. So you take this value, draw it, punch it through the simulator, it gives you something like 4 bursts. Let's say you care about this number 4. And you plot it in the joint distribution in this 2D space, that's parameter data, and that's that value. Because this thing is stochastic, you run it again tomorrow, it might be 5 bursts. You run it again later, it might be 2 or it might be 0, right? So it could take on a bunch of different values, and you take a different value of the parameter, it gives you a different simulation. You can do this, and this is basically what I did, but just with 28 parameters. This cartoon, if you do this with the 1D parameter space, 1D data space, you can, this is what 1 million or like 6 simulations might look like. And then you just basically try to learn this joint relationship with machine learning, right? Specifically with what we use normalizing flow, but you can basically picture like if you had a Gaussian and you had one choice of like where to place the blob to maximize the probability of the Gaussian given all those dots, and that's the parameter you try to learn in your neural network, and that's called a Gaussian mixture or mixture density network. But here we're using flow, which is sort of similar idea with a bunch more complications. But in the end, you get a thing that looks like this, right? It's a contour of probability evaluations, and what you want is all the dots that you had, they should get high probability. Yeah, so then when you have real data, you then can ask the thing that you learn, which is the neural network, and you say, well, given that I had 4 bursts, what is the likely parameter value that I got to produce the simulation? And because, again, stochastic, you want a distribution instead of a single number, so you get what's called parameter posterior distribution, right? Okay, so this is the machinery behind simulation-based inference and neural density estimators. And in this particular case, we're just directly targeting the posterior distribution. And I'm actually kind of running out of time. So again, the plug for the thing, this is also super easy to use. If you have a simulator, just, it's 5 lines of code to get samples that matches simulator outputs with their real data, basically. I mean, if you actually use this with your simulator, it's going to be a little bit more complicated, as I did, because I spent 2 years doing this. But yeah, happy to talk about this with anybody afterwards. But essentially, if you take simulated data that you know what the parameters you use to generate this is, you can punch it through the thing that you trained, and you say, well, give me numbers, and I should expect that it matches the thing that I used to generate the simulation in the first place, right? And basically it does. And then if you take those little black dots, which are the predictions, and you run it through the simulator again, it should give you data that looks like the data you just gave it. And this is all simulated data. And it does that as well. And this is not a cherry-picked example. You can have different kinds of dynamics in the network, and it kind of works depending on what exactly the dynamical regime is. And the nice thing is that then it also reproduces other features of the recording that you didn't tell the neural network to be trained on, right? So it produces, the network automatically produces stuff that's, the PCA is correlated with the temporal dynamic, so it's not super surprising, but it's still nice to see. And then you can analyze the distribution of models that you got out, and you can basically look at correlations between different parameters. So I'm going to skip this because, yeah, right on time. But essentially, different dynamical regimes of the simulated network has different parameter correlations, right? So these are not just single fixed values that instantiates a particular network regime, but it's kind of like the parameters live on this manifold, and everything on this manifold gives you the same dynamic, and then you kind of change to a different surface, and that surface gives you a different dynamic regime. That's the takeaway here. Yeah, so lastly, we applied this to some real data, some recordings from brain organoids, which are stem cell derived brain tissue. Essentially, over the course of development, they have this very distinct feature of population activity, which is that the bursts get shorter and shorter, and this happens in real development as well. And the thing is, we can't really measure a lot of the stuff that you want structurally in the organoid, just similarly as to the brain, right? You can't pick apart stuff in the real brain. So you want is to then, sorry, these are just the developmental trajectories. So what you want is to kind of make a simulated model that looks like an organoid, basically. So using the whole framework as I described to you, find models that basically produces data that looks like this, that changes like this over time, okay? And yeah, so that's what you might call a digital clone. And this is what the real data looks like, long story short. If you do this, you train on the, and the training of this neural network is just from the simulations, right? It has never seen real data ever. But just based on that, now you give it observations of real data that kind of looks like the simulated data. It then produces with you to you, or gives you parameter values that then you can simulate, and it looks like it learned something reasonable, okay? And this is just a summary plot of that. So yeah, which is all to say the model basically gets you values that looks like real data. And then you can interrogate the different model parameters for what mechanisms might have happened or changed throughout the course of development that changed the dynamics. And I think I'm going to skip all this as well because these are very specific neuroscience things. But happy to talk about this later if people have questions. At the end. So this is how we're going to solve this inverse problem. So I guess the thing I want to emphasize is that the neural network there was a black box thing. And typically we think about, and in neuroscience, you want like mechanistic models, right? So this is a way to combine like really powerful neural network-based black box machine learning-based things with real mechanistic models to give you some kind of interpretations. And then finally, you don't have to use this on spiking neural network models. You have a simulator that produces simulations given some numbers. You can do this with any kind of stuff. So like, for example, cognitive models, if you know what a drift diffusion model is, or connectomics, so simulating different wiring rules that gives you how structurally different things connect to each other. And you don't even have to be Bayesian. If you don't like Bayesian being Bayesian, I don't really subscribe to being Bayesian, and there are much more intuitive ways of doing this optimization. So we have this paper now that just got up on arXiv. And then you can also, we are also taking this approach of just like, forget about all this mechanistic simulators, let's just try to make fake data as good as you can, and then try to use it for a bunch of different stuff. So for example, if I know how to make fake data that looks like state A versus state B, I can use it for a classifier training that augments some very sparse data you might have. So this is probably going to go up soon too. Anyway, so that's the summary for today. Out of all things, if you forget anything, I hope you just take away the ideology. And like, if you're analyzing brain data, think about it as like physical measurements from a physical system. But if you're interested in the approach of parameterizing power spectra, or combining mechanistic and machine learning models to basically do this inverse problem, whatever the problem, your problem is, happy to talk more. We're hiring postdocs. Jakob, that's my current boss. Brad is my last boss. Pedro, who is starting his new group, is also hiring. So if you're interested in any of the approaches specifically for neuroscience, happy to chat more. They're all great people. Thank you.
Questions for Richard?
Hi, great talk. Thanks for that. I was wondering the aperiodic component, essentially the slope before the knee and after the knee, they have different interpretations. Yeah. Or any interpretation? I think the most of the work that you presented with the EI balance that was
Is after the knee, yeah.
after the knee.
It depends on your model. So in our old model, there was nothing going on really in the lower frequency part. So there was no interpretation. That's the answer to the question basically. But like physiologically, what I can tell you anecdotally is that the lower frequency components tend to be more dominated by the actual dynamics of the population. So instead of this like synaptic current interpretation, it's actually just the firing rate of the circuit.
So you do a simulation and then you distill it down to a kind of a vector of observables that you then pass on to your neural network model. What are the principles behind which observables you pick? Are you picking them based on what interests neuroscientists, or you're picking them with a view to getting a kind of a complete description that would help your neural network model?
That's a great question. So you can do both of those approaches. The one I'm taking is the first one that you described, which is I know that as a neuroscientist and as the people around me, there are certain things that you care about, for example, like the inner spike interval distribution. So I really care about reproducing this particular feature, sans everything else. So that's the thing I give it to the network. You can care about the power spectrum or specific features of the power spectrum, right? The second thing you described is called sufficient statistics, basically. So how do you distill the simulation, which is, if you look at the spike times, it's like, 1,000, 2,000 neurons by some thousand number of time steps. It's a huge data point. You want to describe this as fully as possible. So for example, taking the number of principal components that preserves 99% of the variance, right? So that's a whole different field in statistics of like distilling these sufficient statistics for this. And there are approaches of learning that jointly with the density estimation network so that you don't have to pick any features yourself, but that you just give it the raw data and it tries to optimize jointly what's the most like informative feature to tell you about the simulation parameters.
I'm curious, like, when you do this feature things, do you observe weird things, weird good parameters, like if you, for instance, add you want to pick the PSD. If you remove it, you have like many good parameters. If you add it, you have one possible good parameter. I don't know if it's clear.
Yeah, like you're asking if adding more data constrains the parameters more tightly, right?
Yeah, I'm wondering, like, I guess you have played a lot with all this stuff, and I'm wondering what's the landscape of the parameter looks like. Does it change a lot between when you change a bit the?
So I haven't done the thing that you're asking. By like successively adding or subtracting different features from the data. But I can tell you, for example, in a theoretical sense, if you give it the entire spike raster and that you're like, that's the thing that I want to reproduce exactly, then it will constrain the parameter to the set that's exactly producing that set of spikes. If it was like simulated spikes. If you took real spikes, you will never get anything because you will never get exactly that spike sequences, right? So that's sort of the philosophical answer to your question, which is you have to do some kind of abstraction to say, these are things I don't care, be a noise or just like it's aspects of the data I don't care about, and then just try to reproduce some other stuff.
Yes, it's related to that. Like once you finish training your machine learning model, can you explore somehow the joint probability distribution of the parameters versus whatever output you wanted to look at, or you can only go back and say, I want to get this, I don't know, interspike interval. These are the parameters that could give me that. Or you can look at the whole.
No, so the machine learning, the neural density estimator has only knowledge about the thing that you gave it. So if you gave it the power spectrum, which is, let's say, 1,000 frequency bins, it only knows that. It cannot produce you any predictions about what might changing these parameters look like for the different.
No, I don't mean that. Like you choose whatever X you want.
Yeah.
Now the output would be the joint distribution of my fixed X and all possible values of
Exactly.
my parameters.
Yeah.
So I thought this question was, is this joint distribution looking strangely somewhere? Like discontinuous somewhere? Is some parameter that's behaving weirdly in this joint distribution?
Oh, that, yes, depending on what exactly the parameter is, it could be bimodal, it could have these islands in the 2D marginal. Yeah, I haven't really even looked at that, but that's sort of the mechanistic interpretation that you want to make afterwards.
In the interest of time, let's move on. I'm sure will be have chance for questions later. Thanks.